Hi guys, welcome back to the channel. In this video, I'll be looking into the genes mutations topic of the A-level biology curriculum. So let's begin. So first looking at the gene mutations themselves. So they are basically a change in the base sequence of DNA or chromosomes. So when we're talking about base sequence, we're talking about the bases thymine, adenine, cytosine, and guanine, and the sequence of these. So in a mutation, the base sequence is basically changed. And these mutations occur spontaneously during DNA replication. So you cannot predict when they will occur, as they just occur randomly. And even though they occur randomly, there are some things called mutagenic agents which can increase the rate of these gene mutations. And for example, UV light or cosmetics, and all of these are also mutagenic agents. Okay, so now looking into the types of gene mutations. So first looking at the base deletion. So base deletion is basically where one or more bases are removed from the DNA sequence. So for example, if this is our original sequence, what happens is there would be a base deleted. So in this case, this thymine is removed from the DNA sequence. So what happens is it changes the base or the triplet sequence from the point of mutation. So if you remember what the triplet is, so triplet is basically three of these bases which would code for an amino acid. So we have TAA to start with, then CT and G, then CAG, and then GT. But what's happened now, even though the first one's still the same, TAA, after that, so when we have CGC, because the T has been removed, we have a new triplet sequence. And this is not just for the next one, because we have removed this T, it causes all of these bases to move to the left, and that's why we have, so from the point of mutation, the whole triplet sequence would be different now. It wouldn't be the same, and we call this a frame shift because we have shifted all of the bases, and in this case towards the left. Okay, so now looking at the next type of gene mutation. So this is base addition. So in this, one or more bases is added to the DNA sequence. So it's basically the opposite of gene deletion, base deletion. So this again changes the base sequence from the point of mutation, and this also causes a frame shift. So if we're looking at the original sequence, so we have the triplets TAA, CTG, CAG, and then GT. After the gene mutations has occurred, we have an addition of the base cytosine. So what this causes, even though the first triplet would be the same, TAA, so that would be coding for the same amino acid, but then if we look at the next one, we have CCT. So we've gone from CTG to CCT, but it's not just this that has been mutated. It's all of the base sequences from this point of mutation are different now. So we had that CAG, so now that's turned into GCA, and we have GGT, which will also be different. So as you can see, this has caused a frame shift again. It's caused all the bases to basically move to the right. So now looking at the final type, which is the base substitution. So in this, there is a replacement of a base by a different base in the DNA sequence. So now looking at the actual sequence, so we have TAA, CTG, CAG, and GT, but we basically, for a T, we have swapped it with a C. So we have swapped T with a C, so it's just a single change. And this would just change one base. So it's just changed thymine, in this case for cytosine, and it would just change one triplet. So if we're looking at the triplets, so TAA, CTG, CAG, and then the other ones. So if we look at every single triplet before and after the mutations occurred, we can see that both of them will exactly be the same, except for the where the mutations occurred. So from the CTG to we have got CCG, and that's the only triplet that's been changed. Because it's only one triplet that's been changed, we're not doing anything to the other triplet. That's why it's not a frame shift. And due to the degenerate nature of the genetic code, the new triplet may still code for the same amino acid, so we may not have any effect on the actual amino acid. For example, if we're looking at valine, we have it can be coded for GUU and as well as GUC. So if there was to be an mutation, a base substitution, where it went from GUU to GUC, we would still be coding for the same amino acid, so that doesn't really make any difference. Okay, so you need to be aware that mutation may produce a non-functional enzyme or a protein. So if we're looking at what happens in a mutation, so gene mutation, so we change the base sequence of DNA, and it can be by base substitution, base addition, or base deletion. So this would change the amino acid sequence, especially with base addition and base deletion, not so much with base substitution, because again, due to the degenerate nature of the base sequence, we might still be coding for the same amino acid, but because we cause a frame shift in the base addition and base deletion, we would almost always be coding for a different amino acid sequence. We have just changed our amino acid sequence due to the gene mutation. So then what happens, because the amino acid sequence has changed, is the specific position of the hydrogen ionic or the disulfide bridges changes. So remembering that when we get into the tertiary structure of the protein, we have all these different bonds, so hydrogen ionic disulfide bridges. So let's say a mutation occurred and it produced this protein. So as you can see, these have different structure because they have different hydrogen ionic disulfide bridges, and the bonds have formed in different places due to the position of the amino acids. And this would ultimately change the tertiary structure of the protein, and it can lead to the active site's shape changing in the case for enzymes. So if we have these two enzymes, so this is the original enzyme that we were meant to produce, but because of the change in the base sequence, we have a different amino acid leading to a different tertiary structure, so we have formed this mutated enzyme. So what this means is the enzyme can is no longer complementary to the substrate, so enzyme substrate complexes can't form. So if this was our substrate, so this was meant to fit to the original sequence, and this would fit, but now because we have a mutation, this can no longer fit into the enzyme, the mutated enzyme, and we cannot form a ES complex, and the protein is therefore no longer functional. And this is how a mutation may produce non-functional enzyme or proteins. Thank you so much for watching this video. If you did like this video, don't forget to subscribe to see more of these, and you can watch my recent videos by clicking on the links popping up. Thank you.